Hello, my name is Vanessa Valencia, and I am going to be presenting on this paper called Efficient Genome Editing for Pseudomonas aeruginosa using CRISPR-Cas12a. So I wanted to go over what the research aim in question was for this paper. So the aim was to explore an alternative option for editing Pseudomonas aeruginosa, and the question that the researchers had was, can the CRISPR-Cas12a system be used to perform mediated genome editing in Pseudomonas aeruginosa? So now I'm going to go over some background. Pseudomonas is a genus of gram-negative bacteria. Pseudomonas aeruginosa is an opportunistic pathogen, and it causes nosocomial infections. It has developed resistance to a number of antibiotics through a range of different mechanisms, such as efflux pump and drug inactivation. So resistant Pseudomonas aeruginosa infections are known to increase in-hospital mortality, the length of hospital stay, and hospital costs. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So CRISPR technology works by being able to identify target DNA, cut it, and then the researchers can edit it. These edits can include deletion, insertion, or modification. The reason why the researchers of this paper decided to investigate Cas12a was because of its potential to edit regions within Pseudomonas aeruginosa that lack G-rich PAM sites, which are typically favored for Cas9. So for this study, the researchers created pCas12a cRRNA plasmid series. So this included pCas12a cRRNAV, PCPF1 RK2T, pcRRNAV, and pcRRNAY. They did list the plasmids that were used in the study in Table 1 of the supplementary section, and then they listed the primers that were used in Table 2 of the supplementary section. So for creating the cRRNAs, they used Cas Designer, and then they used Cas Offfinder to search for and avoid any potential off-targets. The pCas12a lambda red plasmid was transferred into Pseudomonas aeruginosa, strain PA01, through conjugation or electroporation. The transformants were then identified using PCR, and then those PCR products were confirmed using Sanger sequencing. And then they used 500 base pairs as the standard length for the homology arms, and the plasmid pCas12a lambda red and pcRRNAY were cured after they were edited, and then they were confirmed using colony PCR. The Bioscreen C analyzer was used to monitor the growth of the PA01 strains that were harboring different genotypes. So they also did a piocyanin assay, and that was to measure the production of piocyanin of the different strains. And then colonies that escaped editing were subjected to analysis using colony PCR, and they were looking at the target genes. And that also included looking at the FNCas12a cassette and then the cRRNA cassette. So this figure shows the schematic outline of the two plasmid Cas12a mediated genome editing that they used in Pseudomonas aeruginosa, and they broke it down in how many days it took to construct it and then what they did on each of those days. So this figure shows the maps of the pCas12a lambda red and the pcrRNAv plasmids, and this was created using SnapGene 3.1. So on the left, it shows the plasmid map for the pCas12a lambda red plasmid, and then the right shows the map for the crRNAv plasmid. So I'm going to go ahead and go over the results. There were 6 main sections, and then I'll go into the data that backed up these results. So for the first section, what they did was they cultured the PA01 strains for 24 hours after plasmid introduction. So they found that the negative control had around 10 to the 3 colony forming units, referred to as CFUs, and no colonies were observed on the plates with the pCas12a crRNAx transformants. So this data suggests that FN-Cas12a specifically cleaved the genome, leading to bacterial death. It also suggests that the activity of non-homologous end-joining related proteins were not sufficient to repair the damage that was done by FN-Cas12a. So for the second section, the researchers created and used that two-plasmid system, and so they noticed that the number of viable cells was 5.0 times 10 to the 8, and the CFU per microgram was 248 for the pCas12a lambda red harboring electrocompetent cells. For the third section, they wanted to test the capacity of the CRISPR-Cas12a system to do gene insertion and replacement using the RFP and LAC-Z genes. So they found that editing efficiencies were 75%, so that was 9 out of 12, and 67%, which was 8 out of 12, for insertion and replacement of the RFP gene. And they did verification for insertion and replacement for the LAC-Z gene by only selecting blue colonies for analysis from a XGAL plate. And so they found that the number of colonies was 97%, which was 186 out of 191 for the insertion samples, and 95%, which was 123 out of 130 for the replacement samples. But both kinds of editing had 100% efficiency, which was 12 out of 12. And for the fourth result, the researchers slightly modified the CRISPR-Cas12a system to be used for deletion of large DNA fragments. And so when they tested it out, they found that the dual crRNA strategy was more efficient than the one crRNA strategy for the deletion of those large DNA fragments. So continuing on with the results, in the fifth section, serial editing is an important genetic manipulation technique, so they wanted to see if the CRISPR-Cas12a system could be used for this. What they did was they chose the gene clusters PHC A1G1 and PHC A2G2, since they encode for enzymes for phenazine 1-carboxylic acid, abbreviated PCA, and this is an intermediate precursor, and these enzymes call for the biosynthesis of PCA. And they found that all the mutants except for delta PHC A2G2 formed yellow colonies. This indicated that the piocyanin production capacity was lost. This data also suggests that the gene cluster PHC-A1G1 plays a dominant role in the PCA biosynthesis rather than PHC-A2G2. And for the sixth section, the researchers wanted to examine the colonies that escaped the FN-Cas12a Cas12a mediated genome editing. So it is important to know that escape events are common in CRISPR-Cas mediated genome editing. What they did was they picked 4 colonies, 1 from the delta MEX A plate and 3 from the delta PHC A2G2 plate. And then they did PCR analysis on the target genes, which was MEX A and the PHC A2G2. And they found that the loss of the CRRNA cassette or the Cas12a cassette was the most probable reason for these escape events. And this finding is consistent with other previous studies that involved CRISPR-based editing systems. So I'm now going to discuss the data that was presented in the paper. So this figure overall shows the CRISPR-Cas12a based system and how it enables highly efficient gene deletion, insertion, and replacement within the PA01 strain of Pseudomonas aeruginosa. So in portion A to the left, it shows the schematic for the gene deletion, and then on the right, it shows the colony PCR and sequencing results for deletion of PHCM. Portion B to the left shows the schematic for gene insertion, and then the right shows the colony PCR and sequencing for the insertion of the LAC-Z gene at the PHZM site. And then the last portion, which is C, on the left shows the schematic for gene replacement, and the right is the colony PCR and sequencing results for replacement of PHCM with LAC-Z, and all these schematics are the 2 plasmid system. So this table shows the deletion efficiency in the PA01 strain using the CRISPR-Cas12a system. So as I had previously stated in the results section, we can see the pCas12a lambda red plasmid in the first row and how the cell viability is 5.0 times 10 to the 8, and that the CFU per microgram is 248 in sucrose, which is a magnitude higher than the other plasmid, which had 5.4 times 10 to the 7 cell viability and 31 CFU per microgram in the NGSO4, and 2.5 times 10 to the 7 cell viability with 19 CFU per microgram in sucrose. So this table is a summary of the genome editing in Pseudomonas aeruginosa using the 2 plasmid system. I highlighted the portions that I had discussed earlier when the researchers wanted to look at the gene insertion and replacement. So we can see the 100% efficiency in editing for the LAC-Z insertion and replacement, and then the 75% efficiency for the RFP insertion and 67% efficiency for the RFP replacement. So for this figure, in portion A, it shows the schematic of the large DNA fragment deletion using the 2 plasmid system. Portion B demonstrates the genetic map of the target gene clusters for deletion, which were PHCMS and MEX-PHCS, and the purple bars mark the selected target sites. And then portion C shows the piocyanin synthesis phenotypes. So we can see that all the mutants except for delta PHC A2G2 produced yellow colonies, where normal piocyanin production results in a blue-green color, and we can see that in the wild-type streak, which is number 1. So in conclusion, the CRISPR-Cas9 system has been much more exploited than the CRISPR-Cas12a system, but this new system can be just as effective. The authors were able to establish an efficient method for genome editing in Pseudomonas aeruginosa by using this CRISPR-Cas12a 2 plasmid system. And the system can perform insertion, deletion, and replacement precisely, as well as enable serial genome editing. And the CRISPR-Cas12a system can be used in regions with medium to low GC content, which are unfavorable regions for CRISPR-Cas9 editing. So for my thesis, the aim is to determine how genetically altering Pseudomonas aeruginosa using CRISPR plasmids will affect its ability to deal with antibiotic resistance and its virulence factors. So the main question I'm asking is, will genetically manipulating Pseudomonas aeruginosa by knocking out genes PA2897 and PA5157 affect its ability to be antibiotic resistant and impact its virulence factors? So I wanted to discuss the connection between my thesis and this paper. So both aim to genetically manipulate Pseudomonas aeruginosa using CRISPR plasmids, and to examine if there are phenotypic differences in the mutated bacterial strains versus the wild type. And this research allows for us to further understand what genes are or are not involved in the virulence factors and antibiotic resistant abilities of Pseudomonas aeruginosa, and this could be implemented in other bacterial strains, and this can lead to potential for new targets for infection treatment or alternative therapeutics to deal with infection. And both projects aim to contribute to understanding ways that Pseudomonas aeruginosa can be genetically altered and how those alterations impact the ability for the bacteria to be successful in infection through virulence factors.